Okay, if we come back to these stages of X chromosome inactivation, we've briefly covered counting and choice, and now we need to think about the initiation of X chromosome inactivation. So the initiation, of course, is all about turning on exist expression. So as we've mentioned before, that the main thing that's required for X inactivation, in fact, the critical determinant is exist. And so initiation requires exist to be turned on and for it to then coat the inactive X chromosome or the chromosome that will become the inactive X chromosome in cis. In other words, from the very one that it's expressed from. So exist, we know this exist expression can be detected in all female somatic cells for the lifetime of the organism. And so although it's the critical determinant for initiating X inactivation, it is there for the lifetime of the organism. But there seems to be some sort of switch in the dependence on X inactivation on exist itself. So in the very early stages of X inactivation, just after exist has been switched on, there is a critical dependence still on exist. And during this time, if exist is switched off, then it's reversible and you'll go back to having two active X chromosomes. But later in developmental time, when you're in the later phases of X inactivation, or indeed in the maintenance phase of X inactivation, which of course is predominantly what occurs for the longest period of time, then there's less of a dependence on X inactivation. If you, at this stage, delete exist, you can destabilize X inactivation, but you won't immediately result in upregulation of that inactive X chromosome. So, because we know that exist expression is really critically involved at the early phases, but also we know if you turn exist on in most cell types, so if we turn it on, for example, on a transgene that's located on an autosome, we don't find X inactivation ensuing. So there are very few cell types where X inactivation can actually proceed even with exist expression. And so this led researchers to postulate that there must be other competence factors, factors that are required to be found within the nucleus or found in the cell in general, before exist can actually bring about transcriptional silencing. And so I'm going to mention some of these other factors as we come through the next few slides. So after this initiation of exist expression, the next stage is spreading of exist and this silencing that ensues. And so over the years, just like with counting and choice, there have been a very large number of theories about how this might occur. But I think now the prevailing model, and the one that everybody seems to accept, is that there is a progressive recruitment of the X chromosome into a silent nuclear compartment. So what I mean by a silent nuclear compartment is a region where RNA polymerase II is excluded. So the RNA polymerase that actually transcribes genes is removed from this region, and the X chromosome is drawn into this region. So what I'm showing you in the picture along the bottom of the slide is first of all a nucleus that's stained with DNA, a DNA stained DAPI, so we can see the DNA. And you can see two regions that are more densely stained than the others on the top right-hand side. These are two inactive X chromosomes. These particular cells are cancer cells that happen to have two inactive X chromosomes. The second, in the third image, you can see exist expression in green, and you can see these two DAPI dents, or these heterochromatic regions, these bar bodies, are also where exist is found. But this image in between, which is called HMCOT1, is a stain for repetitive RNA, so the expression of repetitive elements. And what you can see indicated by the yellow arrows is that these two regions where these bar bodies are found, where exist is found, are devoid of this repetitive RNA. So then when you look at the pixel profile, so this is when a line has been drawn through the image shown in the merge, the line is shown in yellow, you can see that throughout this region you have enrichment of DAPI, so that the DNA stain in blue, and that's shown by the mound in the graph, and a mound in the exist graph, so that the green line, but you can see that the red goes down, so you see much less repetitive RNA being found in that region. And so this is because we know that one of the earliest events after exist expression is exclusion of RNA polymerase II from this region in which the X chromosome is found, and silencing of the repeats. And so we find that the repetitive the repeats that would sometimes be expressed, the ones that are found in this region are not. So the ones on the X chromosome are not being expressed at this time. And it's a very early event in X inactivation. So this happens, this silent nuclear compartment and the silencing of the repeats actually happens before the genes on the X chromosome are silenced. And we know that the X inactivation spreading correlates with how the genes are actually drawn into this silent nuclear compartment. And I'll show you this in more detail in a diagrammatic form in the next few slides. But here I'd like to mention about some of those factors that are differentially expressed, or that these competence factors which need to be present in the nucleus for exist to be able to bring about silencing. So these factors, some of these factors are SATB1 and SATB2. So SATB1 and SATB2 were found because they were expressed in both embryonic stem cells early post-differentiation, but also in the limited number of other cell types that are actually competent to perform this initiation of X inactivation. And SATB1 and SATB2 are factors that are involved in nuclear reorganization. So in other words, they move the chromosomes around. They help to move the chromosomes around. And so we know that these factors have to be there for EXIST to be able to bring about that silent nuclear compartment. So I'm going to go through all of these things I've just said about the spreading of EXIST and silencing in a diagrammatic form. So pictured here is an X chromosome as it might exist in an embryonic stem cell, fairly open because it's being expressed. And so shown on it in yellow are the repetitive elements. So that would be, for example, the intracisternal A particles or the line 1 elements. These repeats that are found spread throughout the genome. Whereas in green are the genes that are currently being expressed because it's an active X chromosome at this point. Shortly after these embryonic stem cells, or the inner cell mass, is induced to differentiate, then you get a change in how the X chromosome looks and how much space it takes up. We know that EXIST expression comes on, that I'm showing here as a blue squiggly line, and we know that SATB1 and SATB2 are involved. So what happens is within this X chromosome, it's recruited into the EXIST domain. But to begin with, it's only really those repetitive elements, which are shown in yellow here, that have been recruited into this region that contains EXIST. So it's only the repeats that are silenced. This recruitment into that EXIST region contains these purple stars, these SATB1 and SATB2 that I mentioned, these competence factors that are required. But at this early stage, while the repeats have been silenced because they are within this silent nuclear compartment, this EXIST domain, we know that the genes remain on the outside, and so the genes remain active, and I'm showing that here as being green. Then in the final stages, when you reach a somatic cell, we know that now more of those genes have been pulled into that region, pulled into the silent nuclear compartment by SATB1 and SATB2 or other competence factors. And now many of the genes are indeed silenced, and I'm showing that here by being a black gene with a red rim. So once the genes are within the silent nuclear compartment and pulled into the EXIST domain, then they too have been silenced and X inactivation has occurred. What I haven't mentioned up till now in the lectures is that, in fact, there are a small handful of genes that don't undergo X inactivation on the X chromosome. So these genes that escape X inactivation remain outside this silent nuclear compartment in somatic cells. So they are never drawn into the region which isn't transcribed because it's devoid of RNA polymerase II. What we know about these escape genes is that they differ slightly between mouse and human. So here, shown on the left-hand side, is an image where you can see that all of the X chromosome is laid out on the left in the human and on the right in the mouse. And the centromeres are shown in purple. So we know that a human centromere is quite central, whereas a mouse centromere is at the very end, right, at acrocentric at one end. And the genes that escape are shown in orange. So most of the escape genes are in the regions that are shown as PAR, the pseudo-autosomal regions. So in humans, there are two of these regions, PAR1 and PAR2, and in mouse, there's just one of these regions. These pseudo-autosomal regions are the parts that are required for pairing of the X and Y chromosome in cell division. And so these genes that are found in the pseudo-autosomal region tend to have a homologue on the Y chromosome. And so this means that they have one copy that is expressed from the X and one copy from the Y in a male cell, but in a female cell you'll have both copies being expressed from each X chromosome, even if that X chromosome is actually the inactive X chromosome. But there are also a smattering of other genes. You can see orange-coloured genes spread along each of these 2X chromosomes, and there are more in humans than there are in mice. And so these orange escape genes that aren't in the pseudoautosomal regions, they don't have a Y-linked homologue, in which case, if they're not silenced, you'll have twice the dose of them in females than you would in males. And so perhaps these escapee genes that don't have a Y homologue might have something to do with the differences between the sexes. What's important to note at this stage is that all of these genes, whether they're in the pseudoautosomal region or not, all of these escape genes reside outside of that silent nuclear compartment. So the reason that they escape silencing is that they're never drawn into that region that contains the exist cloud and that is devoid of RNA polymerase II. So in the next lecture, we'll think about how X inactivation is established and how it spreads.